Hello, my name is Assistant Professor Dino Spagnoli and I'm the Coordinator of First Year Studies in the School of Chemistry and Biochemistry. This is a video tutorial which will provide assistance in your upcoming laboratory session. I'm here to help you throughout your laboratory session, so when you see me in the lab, please come and ask me any questions or concerns you may have. This video tutorial is concerned with the experiment which is determining the solubility of calcium hydroxide, and you'll see that the next part of the video will go through some of the lab to prepare you for the lab, and also some of the calculations that you'll be expected to fulfill whilst in your laboratory. In this experiment, we'll be determining the concentration of a saturated solution of calcium hydroxide. To do this, we'll be using 3 independent measurements and then comparing our results of one method with another. The 3 techniques we will be doing are techniques that you may have had some experience with already. Gravimetric analysis involves the evaporation of water from the saturated solution and weighing the solid residue that is left behind. Titrometric analysis involves the titration of hydroxide ions against a known concentration of hydrochloric acid. And determination by pH measurements is related to a pH probe that is used to determine your pH of your solution, and therefore you can estimate the concentration of hydroxide ions within that solution. We will need to work as accurately and precisely as possible. Therefore, we will need to determine the possible sources of error that may be introduced into our methods. There is a difference between accuracy and precision. The precision of a measurement gives an indication of how well several determinations of the same quantity agree, whereas accuracy is the agreement of a measurement with the accepted value of the quantity. Let us take the example, for descriptive purposes, that we are throwing darts onto a dartboard. The green dots indicate where we have hit. If we are aiming for the central bullseye on the dartboard, we can see in this first example we are precise because the different throws of the darts are around the same region, but we are not very accurate as we were aiming for the bullseye in the centre of the dartboard. This second example shows that we are indeed accurate because we were aiming for the central bullseye, and you can see that the 5 darts are hitting close to that target, but we're not very precise because the several determinations of the quantity do not agree. This third example shows where precision and accuracy are in agreement. This is because we are hitting that bullseye, which is the accepted value of the quantity, and the several determinations of the quantity all agree. It is the aim of the chemist during an experiment to minimize errors, and these errors could be imperfections in the technique or apparatus. However, errors cannot be completely eliminated, so therefore we need to be aware of these errors associated with a measurement. And there are 2 main types of errors that could be introduced in an experiment. The first is a systematic error. These remain constant or vary systematically for a series of measurements. So an example would be a 10 ml pipette actually measures 9.98 mils each time a measurement is delivered. Or there are random errors. These vary in sign and magnitude from measurement to measurement. So due to the fact that there are human errors involved in every measurement, we might perform a 10 ml pipette of a certain liquid, however the samples that are delivered will vary in volume. I'd now like to just go through very briefly some little tips and hints about this particular lab. In this lab you really do have to work efficiently. Time management will be key in getting all 3 experiments complete in time. When you are thinking and doing the experiments, you need to identify the sources of errors. And then obviously, being a physical chemistry laboratory, you need to have the correct calculations and always remember units. Okay, you'll be deducted grades if you do not put the correct units in. And then finally, think critically about the estimates of the solubility using the three methods. Are the estimates in line with each other? Are they precise? How accurate are they? Where are the sources of error? Okay? If you have any troubles, just come and speak to me or your demonstrator, and I will speak to you and see you in labs.